Hello and welcome to lecture 3 of our course on stochastic modeling of biological processes. This is the third and the last lecture discussing chapter 1 of the lecture notes. In lecture 1, we covered the first 14 pages. In section 1.1, we discussed a stochastic model of degradation and showed how we can design an event-based SSA which was more efficient and accurate than the naive SSA. In section 1.2, we considered a simple system of two chemical reactions, production and degradation, and presented the SSA A3D3, which we will further generalize today as the Gillespie algorithm. We also discussed theoretical analysis of stochastic models using the chemical master equation. In lecture 2, we included a discussion how we can implement the SSA A3D3 as a MATLAB code. Today, we will discuss the second half of chapter 1, covering sections 1.3, 1.4 and 1.5. We will present the Gillespie algorithm capable of simulating arbitrary systems of chemical reactions. We will also discuss additional methods for analyzing the chemical master equation. Let us consider a system of Q chemical reactions. Our illustrative example on page 26 includes 4 chemical reactions. So Q is equal 4, and it models two chemical species denoted A and B. The first reaction is the dimerization, where two molecules of A react to produce some products, which we do not explicitly model. So we denoted them as this symbol, empty set, which denotes all unimportant chemical species we are not interested in. In the second reaction, one molecule of A reacts with one molecule of B to produce some products, while the third and the fourth reaction are production reactions. To develop a stochastic simulation algorithm, we need to introduce a probabilistic definition for each reaction. As in lecture 1, we will do this by assigning a propensity function to each reaction. It is a function alpha I such that alpha I times dt is the probability that the ith reaction occurs in the infinitesimally small time interval t plus dt. We already know what are the propensity functions for production and degradation reactions. For all other reactions, we can find them in tables on page 16 of the lecture notes. Looking at these tables, we obtain the following propensities for our illustrative system. The third and fourth reactions are productions of chemical species A and B, which we studied in our last lecture. Therefore, we already know that the corresponding propensity functions, alpha 3 and alpha 4, are constants, products of the rate constant and the system volume nu. Considering the second reaction, bimolecular reaction A plus B is going to some products, its propensity function is proportional to the number of available pairs of molecules A and B, which is the product of A times B. This is multiplied by the rate constant, which has units of volume per time. This scaling with volume is again natural. If we make the reactor smaller, then the propensity increases, because any pair of molecules is more likely to collide and react in a smaller reactor. Considering the first reaction, when A reacts with itself, its propensity is again proportional to the number of available pairs of reactants, which in this case is A times A minus 1. In particular, if there is only one molecule of A in the system, then this reaction cannot occur, and its propensity is 0. Once we define the propensities, it is easy to write the SSA for simulating a general system of chemical reactions. It is given on page 25 as the Gillespie SSA, denoted SSA A5D5. It is a generalization of the SSA A3D3, which we discuss in lecture 1 and implemented in MATLAB in lecture 2. At each iteration, we ask two questions. When will the next reaction occur? And which reaction takes place? Each iteration of the Gillespie algorithm is written in terms of 4 steps. First, We generate two random numbers R1 and R2 uniformly distributed in the interval 0, 1. In step B5, we compute the total propensity function alpha 0, which is the sum of propensity functions of all reactions. In step C5, we answer the first question: when will the next reaction occur? The time to the next reaction event is exponentially distributed with mean alpha 0, so it is computed as 1 over alpha 0 logarithm 1 over R1. Our second question, which reaction takes place, is answered in the last step D5. We use the second uniformly distributed random number R2 to select the corresponding reactions, because we know that the relative probability of the jth reaction occurring is its propensity function alpha j divided by the total propensity function alpha 0. Then the jth reaction takes place, therefore we update the numbers of reactants and products of the jth reaction. In lecture 1, we considered a simple chemical reaction example, the production-degradation system, with two reactions. Substituting their propensities in the Gillespie SSA, we obtain the SSA A3D3, which we implemented in lecture 2. In this lecture, we consider a more complicated example of two chemical species, A and B, which are subject to four chemical reactions. The Gillespie SSA is applied to this system on pages 26 to 29, with our results presented in Figure 1.5. Figure 1.5 has four panels. The first two panels, A and B, show the results of 5 realizations of the Gillespie SSA. The number of molecules of chemical species A is plotted on the left panel, and number of molecules of the chemical species B is plotted on the right panel as functions of time, as solid lines. Different colors correspond to different realizations. Each realization is started with no molecules of A or B in the system. We observe that some molecules of A are produced, then some are removed, but after some time, the number of molecules of A fluctuates around the value of 10. Our chemical system has four reactions, written here. To use the Gillespie SSA, we assign the corresponding propensity function to each reaction, as given here. We can also write its classic deterministic ODE description. If you don't know how to do this, there is Appendix A on page 293 discussing how we can write an ODE description for a general system of chemical reactions. In the case of our chemical system, its ODE description is given here. It is a system of two ordinary differential equations for concentration of chemical species A and B, which we denote by lowercase letters. Our second order chemical reactions are removing molecules from the system, so they correspond to these negative terms. The production reactions are corresponding to these positive constant terms, K3 and K4. Multiplying the concentrations A and B by the system volume nu, we obtain the number of molecules in the system, which we plot as black dashed lines. We use the same initial condition as for the stochastic model. That is, we have no molecules of A or B initially in the system. Although our picture could suggest that the black dashed line could be a description of the average behavior of the system, this is not exactly true for this model. In fact, if we average over a long time realization of the Gillespie algorithm and ask the question, what is the average value? The number of molecules of A eventually fluctuates around, we obtain 9.6. And we obtain 12.2 for molecules of B. These values are not equal to the stable steady state of the ODE system, which is 1010 for our parameter values. In particular, the deterministic ODE system does not describe the average behavior of the stochastic model. One could argue that the differences between 9.6, 12.2, and 10 are relatively small, and the OD system could still be considered a reasonable approximation for this model for our parameter values. However, this is not the case in general. In Chapter 2, we will show that the difference between deterministic ODs and the average behavior of stochastic models can be significant. In addition to the average behavior, stochastic modeling can give us extra information, like information about fluctuations or correlations. For example, if we look at this blue realization, then we can observe that the number of molecules of A is below the average and the number of molecules of B is above the average. On the other hand, if you look at this red realization, then it is the other way around. The number of molecules of A is above the average and the number of molecules of B is below the average. We can further investigate this possible correlation by plotting the stationary probability mass function, the stationary distribution, which is shown in panel C of figure 1.5. We can see this negative correlation, which is caused by this reaction, where A and B can react and be removed from the system. So if there is more A, then more B will be removed from the system, and vice versa. We can also sum the two-dimensional stationary distribution to obtain marginal distributions, like the marginal distribution of A, which is plotted in panel D, where I highlighted the deterministic ODE mean value 10 as the red bar. The stochastic model can give us more information than ODEs. It can give us the whole distribution of A plotted here. Therefore, figure 1.5 illustrates that we can obtain any information about the system which we want by applying the Gillespie algorithm and visualizing the computed results accordingly. So let us now summarize what we can do for general chemical systems of n chemical species A1, A2 until An that are subject to Q chemical reactions. In our illustrative example, we had two chemical species and four chemical reactions, so capital N was equal to 2 and Q was equal to 4. We used the Gillespie algorithm to simulate it, which we stated in its general form as the SSA A5D5 on page 25. In particular, if we consider an arbitrary system of chemical reactions, we can still apply the SSA A5D5. To use this algorithm, we have to specify the propensity function of each chemical reaction, which are given in tables 1.1 and 1.2 on page 16. If this course was being taught to students of biology, then the emphasis would be on algorithms and their implementations. They would be focusing on solving complex biological problems using the algorithms from the lecture notes. So, for well-mixed chemical reaction systems, they could just stop here with the Gillespie algorithm. However, this course is offered in the Mathematical Institute and it is not only about programming. Our aim is to understand the mathematics which can be found behind the algorithms from the lecture notes. So, how do we analyze stochastic models of general chemical systems? In our previous examples of their analysis in lecture 1, we always started with the chemical master equation, which is equivalent to the Gillespie algorithm. For a general system, it can be written in the following form. It is actually in the lecture notes later as equation 378 in chapter 3 because we decided it is more illustrative to work with simple examples in chapter 1. I have put it here just to highlight that the chemical master equation can be written for any system of chemical reactions. It is usually a very large system of ODEs. In fact, if there is a production reaction included in the system, then the chemical master equation will be a system of infinitely many linear ODEs, as we have already seen with our production degradation system in lecture 1. The chemical master equation is the equation for the joint probability mass function P and T, which is the probability that there are N1 molecules of A1, N2 molecules of A2 until Nn molecules of An in the system at time T. For each reaction, there are two terms on the right-hand side, the gain term and the loss term. So there are 2 times Q terms on the right-hand side, where Q is the number of chemical reactions in the system. Therefore, we can always write the starting point of the mathematical analysis, the chemical master equation. So the next question is, can we use it to obtain some interesting results? The answer is, we sometimes can. We could try to solve the chemical master equation numerically, but we will not discuss such an approach here. We will instead focus on a pen and paper approach, discussing simple stochastic models where we can obtain some analytical results. Such simple systems are building blocks of large, complicated models of biological systems. Having analytical results for them is actually very useful, as we discussed in the motivating lecture of our course, in lecture 0, because we can use them to benchmark our computer codes. That is, we can use them to check whether there are any errors in our computer implementations by comparing the computed results with exact analytical formulas. We have already presented some analysis in lecture 1. We considered chemical systems containing one chemical species A, and we denoted the mean and variance of the number of molecules at time t by these formulas, where P and T is the probability that AT is equal n, that is, that there are n molecules of A at time t in the system. We discussed two approaches to find MT and VT. In the first one, we first solved the chemical master equation to find P and T, and then used 112 to find mean and variance. This was the approach which we used for the degradation reaction. In the second approach, we first derived ODs for mean and variance by manipulating the chemical master equation. This was the approach which we presented for the production and degradation system. The approach 2 is applicable to any system containing only first-order reactions, like degradation, and zero-order reactions, like production with a fixed rate. Then we can show that the evolution equation for mean is equivalent to the standard deterministic ODE description. This is not the case when the system contains at least one second-order reaction, like the dimerization reaction, as we have already seen in our previous example. As an exercise, you could try to apply the approach to yourself to such a system, to confirm that you are not able to find closed equations for the mean and variance by summing up the chemical master equation. Today, we will discuss an approach for finding the mean and variance using the probability generating function. This approach could also be considered as a subset of approach 1 because it can give us the probability mass function P and T as well. Consider a chemical system containing one chemical species A. Let P and T be the probability mass function. That is, P and T is the probability that there are n molecules of A in the system at time t. Then we define the probability generating function by this formula. If we know the probability generating function, then we can obtain the mean m by differentiating it with respect of x and substituting the value x is equal 1. The variance v and the probability mass function p and t can also be obtained by differentiating the probability generating function. Formulas 133 till 137 are derived on pages 19 to 21 of the lecture notes. In Oxford's, students taking our course also took a basic course on probability in their first year, where formulas 133 till 137 are derived. Therefore, I am not rederiving them in this video. For some systems, we are also interested in the stationary distribution, which can be obtained as the limit of P and T as time t goes to infinity. The stationary distribution is related to the stationary probability generating function, which is the limit of gxt as time t goes to infinity. Formulas 133 till 137 for the time-dependent case naturally imply the corresponding formulas for the stationary case. The stationary mean, ms, which is defined as the limit of m as time t goes to infinity, is just the derivative of Gs evaluated at 1. The stationary variance and the stationary distribution can also be calculated by differentiating the stationary probability generating function. Next, let us show how we can use the probability generating function to find a formula for the stationary distribution. We consider a production and degradation example. In lecture 1, we obtain the following stationary chemical master equation. First, we use the stationary chemical master equation to derive an equation for the stationary probability generating function, which is defined as this sum. To do this, we multiply the stationary chemical master equation by x to power n and sum over n. We obtain a very simple ordinary differential equation for Gs. To solve this ODE, we use that the value of Gs at 1 has to be equal to 1. Then the ODE has the unique solution given as this exponential function. Since we were able to find the stationary probability generating function Gs, we can differentiate it to obtain the stationary mean, stationary variance, and the whole stationary distribution. In particular, we rederive that the stationary mean is just the ratio of rate constants k2 nu divided by k1, and the stationary distribution is just this Poisson distribution, obtained by differentiating the stationary probability generating function Gs n times. It is the Poisson distribution with mean ms. Therefore, the stationary variance is also equal to the stationary mean. The derived Poisson distribution is plotted in figure 1.2 on page 10. It is this red line. Since we know that this is an exact result, this analytical formula, this red line, is a very good benchmark for the computer code I ask you to write in lecture 2. Your computed result, this gray histogram, should perfectly match the red line, provided that you average over sufficiently long time evolution computed by the Gillespie algorithm. If you see a visible error, you should increase the length of your simulation and the error should be smaller. If not, then this would indicate that there is a mistake in your computer code. Another example showing how the probability generating function can be used to obtain analytical results for the stationary distribution is presented in section 1.4 of the lecture notes on pages 19 to 23. Section 1.4 follows the same approach as we did for the production degradation system, but the resulting ODE for GS is slightly more complicated to solve. In section 1.1, we solved the time-dependent chemical master equation and obtained the binomial distribution 1.7 for the probability mass function P and T corresponding to the degradation reaction. For the production degradation system, we have so far only analyzed its stationary properties. So the next question is, can we solve the time-dependent chemical master equation for the production degradation system analytically? The answer is yes. The solution will of course depend on our initial condition. Using our initial condition A0 is equal to 0, it is left as exercise 1.6. To solve it, you can use the chemical master equation to show that the time-dependent probability generating function gxt satisfies a first-order partial differential equation, which can be solved analytically using the method of characteristics. The probability generating function can help us to analyze some systems with second-order reactions. In the lecture notes, we included the dimerization example, where one chemical species A is subject to two chemical reactions given here. Starting with no molecules of A in the system, some molecules are produced by the second reaction, then some are removed from the system by the first reaction, but after some time, the number of molecules of A fluctuate around its average value. The dashed line is the solution of the corresponding deterministic model, which is given by this ordinary differential equation for the concentration of chemical species A. Both panels of figure 1.3 look very similar to the results computed for our production degradation system, but there is one important difference. In this case, the deterministic ODE model does not describe the average behavior of the stochastic model. Using the stationary probability generating function, we obtain that the exact value of the mean is close to 10.13, which is not equal to the steady state of the deterministic ODE model, which is 10 for our parameter values. As an exercise, you could use long-time stochastic simulation to verify that the mean is close to 10.13. Obviously, the difference between 10 and 10.13 is relatively small, so one could argue that the ODE does provide a reasonable approximate description of the average behavior of the system. However, this is not the case in general. In our next 4 lectures, we will discuss Chapter 2, where we will show that the difference between deterministic ordinary differential equations and the average behavior of stochastic simulation algorithms can be significant. We will present chemical systems which cannot be modeled by ODEs. We will discuss questions which cannot be answered by ODEs. And this brings me to the end of our discussion of Chapter 1. We covered the last 3 lectures. I presented the material in a different order, but we covered all main points which are there. Please read pages 15 to 29 of the lecture notes before watching the video of our next lecture. Thank you for watching video lecture of Oxford course on stochastic modeling of biological processes.